Hello, everyone. I see that my video is not working, but I hope you all hear me. Maybe you give me a thumb, so I'm sure that you can hear me. Okay, thank you. Thank you very much. Hello, my name is Frank, and I'm the father of Alexander and the partner of Daniela. Daniela is my wife, and both Alexander and Daniela are living here with retinitis pigmentosa, a condition that has enormous impact on our daily life and for which sadly there is still no solution to restore proper vision. Through patients groups here in the Netherlands, I came into contact with Dave Hogevorst, and Dave is the father of three daughters, and all of three of them are also live with this cruel disease. As parents, Dave and we are deeply involved. We talk to each other often, and we stay closely connected with our professor, Professor Camiel Boon. When my son was first diagnosed, he was only 4 years old at the time. I immediately joined several social media groups on Facebook and wanted to learn everything I could about ongoing research and about how doctors and scientists around the world were working together. I didn't want to miss anything because the sooner there is a progress, the sooner it can make a difference for patients like my son. Along the way, I realized that compared to many people around the world, I was receiving a lot of valuable information from the doctors in the Netherlands. And Professor Boon also explained me that in the Netherlands, we are actually quite advanced when it comes to developing gene therapies. When Dave and I realized this, we both felt a call to share the knowledge we had with others. That is why we asked Professor Boon if he would be willing to organize webinars so he could tell us more about his research and the latest developments. Our only purpose is to make sure knowledge is shared because that benefits everyone. Last year, we held the first webinar, and this year we are happy to host the second. This time, we especially want to thank Professor Boon to tell us more about gene-independent therapies, treatments that might become available in the near future, and that all of us could potentially benefit from. Because in the end, we all need a little bit of hope to help us cope with this terrible, cruel retinitis pigmentosa. We are deeply grateful to Professor Camiel Boon for taking the time to be here with us. Professor Camiel Boon, for the one who doesn't know him, he is a professor of ophthalmology and consultant ophthalmology at Amsterdam University Medical Center and Leiden University Medical Center in the Netherlands. At just 36 years of age, he was appointed full professor, and today he's a world-renowned expert on retinal and genetic eye diseases. Please welcome Camiel, and also I want to tell you that if you want to ask a question, we have a message box, so you can also type questions, and we've received a lot of questions already, and a few of them we will ask also to Camiel. But for now, I give the word to Camiel and hope it will be a very interesting meeting for all of you. Thank you.
Thank you, Dave. Yeah, so a warm welcome to you all. My name is Camiel Boon. I'm sorry that it took a little bit more time because I muted myself again because I had some residual cough from last week when we had to unfortunately cancel because I couldn't speak for more than 3 sentences in a row before some ferocious coughing would erupt. But it's great that many of you have still found the link and are able to join us here tonight for Europe. But I know that there are quite some people from even outside Europe who are joining and will also be joining later. And that is why we're also doing this presentation in English. And well, I'm a professor of ophthalmology and clinical ophthalmogenetics in the Netherlands, in Amsterdam UMC, and also with a small residual appointment in Leiden University Medical Center. And in the next, let's say, 45 minutes or so, I would like to tell you something about the fight for sight, as I would call it, the way we are trying to develop treatments, not only here in the Netherlands, but also in other places around the world. And I would like to give you an update on the new developments, both in genetic, but also in non-genetic therapies for hereditary retinal diseases, such as retinitis pigmentosa or Stargardt disease, or several other diseases like cone dystrophy. So now I'm going to advance my slide. So I don't have any financial conflicts. I'm not sure why there is this green stripe in my slide, but I don't have any financial conflicts to disclose. So I'm not affiliated to any of the companies that are developing gene therapy. What I do like to say is that, of course, for people, for patients with these hereditary eye diseases, but certainly also for me and my colleagues as ophthalmologists, it is really a wonderful time that finally we are able to treat the first of these many genes. So we are trying to treat the first patients with gene therapy. And there's still a very long road to travel that I'm going to talk to you about, but it is really amazing that finally, for the first time ever, we are able to have some conversations in our consulting rooms to say, okay, we might have a treatment for you. And I'm going to tell you about how we came that far, how much longer we have to travel, and what are the promising aspects that are down the road. So in my talk, I will tell you generally about where do we come from, very shortly, some history, then what is currently the state of the art, at least here in the Netherlands and in many other places. What are the current developments in terms of treatments, both genetic and non-genetic? And what are the challenges? I think it's very important to also give you a reality check, which is often, well, I would say underestimated and under emphasized, I would say. And where are we going? What are the most promising fields? Those will take some time. I also want to start with a disclaimer. So if you think that at the end of the presentation, I will have told you everything about your personal gene and disease and where we are going, that will probably not happen. We are going to address as many topics as possible to also appeal to as many of you as possible, and also ask some of the questions that you have submitted previously and that you can also submit in the chat. We have to do some kind of selection because he had more than 200 people subscribing and also many, many questions. So trying to cherry pick the questions that are maybe most applicable to most of you. So just to tell you a bit about the history, I would like to tell you something about this brilliant German scientist, Hermann von Helmholtz, because before 1850, we're actually not even able to look into the back of the eye and to see the retina, because that's what we're talking about in this webinar, hereditary diseases of the retina. But this brilliant German scientist, Hermann von Helmholtz, he developed the ophthalmoscope, which was a way with candlelight, which was extremely difficult, by the way. It was a way to look through the pupil, a small pupil actually. Nowadays, you will probably, most of you know it, you get drops to widen your pupil, which makes it much more easy to look through the gate of the eye. But then they had to look through a small pupil by candlelight with an ophthalmoscope. And that was the first time when we were able to see the retina and all these diseases, including, for instance, retinitis pigmentosa, that could also affect the retina, but also retinal detachment, etc. So it took decades and even longer than decades for all these diseases to be acknowledged. And it took even longer, actually, until, let's say, the 1970s to realize that several different genes are involved in several of these diseases, which is going to be very important. And I'm going to tell you later about that. Our own Dutch hero in ophthalmology, FC Donders, Professor Donders, he actually recognized very early on that this was a huge research discovery, and he immediately improved this ophthalmoscope so that we could also use it in the Netherlands. And actually, he and his students were the first to describe the disease retinitis pigmentosa in 1853 in the PhD thesis of Adriaan van Tricht. And that was extremely difficult to see because you had to look through with candlelight through the pupil. And this is actually the first drawing ever of retinitis pigmentosa in this thesis, drawn by this PhD student himself, who actually had a father who was a surgeon, who was also an amateur, a very good amateur painter. And this was the same for Adriaan van Tricht. So luckily for Professor Donders, he had a PhD student who was not only able do research well and to look very carefully through this with a new instrument through the eye. But he was also a very good artist, so he painted the first pictures, such as this one, of many retinal diseases, including retinitis pigmentosa. And then, of course, we have to talk about DNA because we're talking about hereditary diseases. So just to fresh up your mind, the DNA, our genetic code, enables us to express all kinds of important life functions, all important life functions, actually. So the DNA is the code that makes sure that, for instance, we can also make the right proteins to use to develop an eye and to use this eye correctly and to see the light. And it is very interesting to see that from the 1990s onwards, when the first DNA analysis techniques to look into families with hereditary conditions, first and foremost retinitis pigmentosa of RP, these first techniques were developed. And from then onwards, you can see that very rapidly, with also new techniques kicking off, many of the causal genes for these hereditary diseases were found. And initially, people thought there are only just a few genes are going to cause RP. And we talk only RP, for example. We did find that, for instance, Stargardt disease is caused by just one single gene, but we also have found that retinitis pigmentosa can be caused by more than 100 different genes, which also indicates how complex the function of the eye is. So I personally fell in love with the eye, so to speak, when I first looked through a slit lamp at the iris, so the beautiful colored part of the eye in front of the eye. But soon I realized how wonderful the structure and the tissue of the retina is. So the retina that we're talking about, which can have these genetic retinal diseases. Oh, I'm sorry, the slides are advancing by themselves. The retina is the neuronal lining at the back of the eye, and the retina is very important in first and foremost catching the light and also changing the light into an electrical pulse that is then through the optic nerve brought towards the brain. And this is actually so where the miracle of vision is starting. So light comes into the eye, is refracted through the cornea and the lens towards the retina. And the retina, as you can see it here, actually changes a light particle into an electrical pulse. And the center of the retina may even be the most important part that you could argue, because the center of the retina, or the macula, is the part where most of the cones are. And the cones are important in seeing sharply. So being able to see contrast, recognizing faces, color, etc. But the peripheral retina, as many of you know, of course, is equally important for the visual field and for night vision. And that's where things go wrong in retinitis pigmentosa. So after the light is caught up into the retina, the light is transferred through the optic nerve towards the brain, and that's where real interpretation of vision starts, where you get feelings with what you can see, etc. And you see form, you see movement, etc. So why do I tell you this? Well, because I think I want to make small ophthalmologists out of you, that you especially are very aware of how all these problems with the retina, especially in hereditary conditions, start. So here you see a schematic difficult drawing of how these photoreceptors, the cones and the rods, are embedded in the pigment layer of the eye. And this is a very complex situation. This is even an oversimplification with all the steps that are needed. And what you can see is that in all these steps of vision, which is a very complex situation, there can be mistakes in the genetic code. There can be mistakes in the DNA, and all these DNA mistakes, these genetic disorders, can cause a retinal degeneration. And more than 100 different genetic factors are known with retinitis pigmentosa. So when talking about retinitis pigmentosa, on the left you see what we see as an ophthalmologist, with the blood vessels and the macula and the optic nerve. That's this round area that is normal, and the scan below, the scan that we make often those of you who are also having these problems, you know what scans we make. And we can compare it to the right, where there's a patient with RP with retinitis pigmentosa, and you clearly see that there are pigmentary abnormalities that are caused by the degeneration of the retina. And that's why there is progressive tunnel vision. And in the end, in some unlucky persons, also the central vision can go down quite a lot, which is, of course, a dramatic disease, as many of you know. But fortunately, in the first decades, the central vision can still be quite good. But of course, losing your peripheral vision and this tunnel vision can be extremely invalidating. And this is very schematically for those who want to have an impression, who don't have RP, hopefully in themselves, who want to have an impression of what people with RP see. It's impossible to, of course, depict, but you have this tunnel vision where you have only central vision left, which can already also progressively become blurred. On the other hand, for instance, there are hereditary retinal diseases where the central retina, the macula, is degenerated. So these are patients who do not have night blindness in general. They can, but often they don't, and don't have tunnel vision. But within a few years, their central vision can become compromised. For instance, in Stargardt's disease or X-linked retinoschisis, Best disease, and that's when people start experiencing blurry lines, decrease in central vision, difficulty recognizing people, and this can also, of course, have dramatic influence in daily life, in social life. And these people can then, as depicted here, also have a lot of problem in recognizing faces and reading. So it brings us to what's the current state of the art when it comes to these hereditary retinal diseases, their diagnosis, and also potential new treatments. So I think it is very important when you have such a rare disease, because, for instance, retinitis pigmentosa occurs in about 1 in 3,500 people in overall, I would say, and Stargardt disease 1 in 10,000 it is estimated, that if you are confronted with such a disease, it is very important to have a good diagnosis. If there's a therapy, of course, access to therapy, but this is also, that's still in a small group. You want to have access to genetic testing, because if you know which gene it is, of course, it's you first of all have a clear diagnosis, and you also know, for instance, what the risk is that your children will inherit it. And this will also allow you to, for instance, be eligible for treatments. So it can also help in genetic counseling. And if people want to avoid children to get the disease, you could even have pre-implantation genetic testing to see, which also involves an IVF trajectory. So it's very intense, but it could also lead to preventing your offspring to get the disease. Of course, when you are already poorly sighted or you are already confronted with visual function problems, you could also have low vision support and professional support to be able to be supported as much as possible to continue the things that you want to do and you like in social life, in professional life. Psychological support, social support, this is all important. And that's why I think it's very important to be seen in an expertise center, a center where people really know what they talk about and where multiple disciplines, for instance, if there's also hearing loss in Usher disease, sorry, the slides seem to advance spontaneously. I thought I switched that off. If you also have hearing loss, you can go to an oral doctor. So the more and more there are expertise centers with multidisciplinary care where there are also geneticists you can talk with. And it's very important, I think, that people with these diseases are connected to such centers. And of course, it's very important to be involved in research if you would like to be involved in research and to be close to new developments if there potentially is a treatment for you, or if you can take part in clinical trials, such as gene therapy trials, which I'm going to talk about more. Because finally, there are the first treatments at the horizon of these hereditary retinal diseases. And I think this is really groundbreaking and is extremely motivating. And why is the eye at the forefront of these developments? Well, as you all probably realize, the eye is a very small organ. So you don't have to apply a lot of these experimental medications, such as gene therapy, to already have an effect. And you can only administer it in the eye. So you need only a few drops of this medication, and there's much less risk of problems in the rest of the body because of administration of this new medication. Also, we are able with microsurgery, I'm also performing these surgeries myself, with tiny injections, you can be very meticulous in delivering it at the right place. And also, as ophthalmologists, we are lucky to have very a lot of ways to, even at the micrometer level, evaluate the structure and the function of the retina before and after treatment. So we are really good at monitoring the disease and also the potential impact of treatment, although this is still quite challenging. I will tell you a bit about that later. And finally, the eye is also at the forefront of these new developments, such as gene therapy, because the eye is relatively unlikely to have a lot of inflammation, a lot of problems after administering a treatment. The eye is immune dampened, so to speak. So the immune system of the eye is relatively inactive compared to the rest of the body, which also helps. Now, we are slowly going towards treatment, and I would like to stress as a reality check that the possibility and the choice of future treatments will very much depend on which type of disease you have, what the underlying gene is, if we can find it, because right now we can find about 50 to 75 or 80% of the causal genes, depending on which type of retinal dystrophy you have. For instance, in RP right now, we can find about 75 to 80% of the genes, and the rest we cannot find them. So these people in whom we cannot find the gene are by default not eligible for gene therapy at the moment, unfortunately. So we really have to step up efforts to find all these genes. And the possibility and the choice of treatment will also depend on the stage of the disease. We'll tell you a bit about that later. So if you have very advanced disease, you might not be eligible, for instance, for gene therapy, but maybe for other diseases and vice versa. And of course, most importantly, it depends on what your preference is, because even now there are emerging some treatments that are also gene independent. And actually, we're going to start with a trial, an RNA therapy trial in Amsterdam for patients. And some of those patients are actually already in another trial, so they will have a choice which treatments they want to have in a trial setting, which is also confusing and difficult and makes it very important for me to explain very well to these people. Okay, these are the pros and the cons of treatment A. These are the pros and the cons of treatment B, and both of the treatments we're not sure. We're not sure if they will work and which one will work best if they work. So we're also entering a more complex era when it comes to treatment, although I must stress that many of these types are still not treatable. And when it comes to the approaches to the treatment of IRDs, I think very broadly we can distinguish between gene or disease specific approaches and gene or disease independent approaches. And I first want to talk with you about the gene specific treatments. So what about them? Well, there's quite a lot going on. For instance, in Amsterdam, we're taking part in several first ever phase 2 and 3 therapeutic trials for retinitis pigmentosa, but also we're going to start a trial for Stargardt's disease. And that is really nice. It's also very challenging, not only first and foremost for the patients who are involved, because it takes much more time than just a checkup in the clinic, which is already, as some of you know, time consuming. But taking part in these trials, you need to jump through so many more hoops in terms of evaluation. That is very intensive for the patient, for the family, but it is also intensive, I must say, for our department and our trial center. So, for instance, in the Netherlands, we have 17 million inhabitants, almost 18, I think, but there are only 3 highly equipped trial centers for these trials, including Amsterdam. So, because you need a very good infrastructure. So I'm going to discuss 3 key genetic therapy approaches with you in this webinar. And the first, I would say the gold standard that has been used thus far is the so-called AAV-based gene augmentation. Very difficult term. I'm going to explain what it means. So AAV means adeno-associated virus, which is, you could say the envelope. So it's like an almost harmless virus that is almost empty. So you can put the right gene that was missing. So in a person who misses a certain gene, for instance, causing retinitis pigmentosa, you can put in the right gene with the right signal in these virus envelopes, and you inject through a very small cannula, as you can see in the top left. You can inject a cocktail of billions of these viruses under the retina, or maybe in the future just in the eyeball, which will be an easier operation, but it has its disadvantages at present. You inject them under the retina, and this cocktail of viruses, the viruses know where to go. They know how to infect, so to speak, the photoreceptors, the cones and the rods, which are missing the gene. So they nestle themselves into the core of the cells where the DNA is, and they put in the right DNA, and hopefully, that's what we're testing now, they will express the gene again, and the cells that are still alive will hopefully be rescued, or most of them will hopefully be rescued based on these viruses that deliver the correct gene. And this is why it's called gene augmentation. So the gene is not working or almost not working, but you give a boost to these cells who get the right gene again using these virus envelopes. And this has been tried already. This is, for instance, already used in the first ever approved gene therapy, which I'm going to talk about a bit later, which is Luxturna for the RPE65 gene, which is a very rare cause and causes retinal dystrophy in only less than 1% of the patients. But still, it's a milestone. Here it is, the RPE65 gene. This was the first ever approved gene therapy by the FDA for any disease. It's very expensive, by the way. I think it's about 350,000 euros per eye, which you can also fill a whole webinar about healthcare costs. But anyway, this is able to really put on the light again in this specific patient group. And this was also approved by the Dutch regulators and the Dutch Ophthalmological Society, but under very strict circumstances, of course, also because of the cost. And they use this AAV-based gene augmentation. There are several other trials going on. We're also involved in that. Last 2 years, I operated on 5 young men with X-linked retinitis pigmentosa due to the RPGR gene, both eyes within 4 weeks, which I think was quite exciting. And I think it hasn't been done before, but that was what's prescribed by the pharma company. So, but this went quite well. So actually, in this pioneering phase, we're seeing that it seems to have a relatively good safety profile, but we have to be very vigilant because this is the first time ever that we're doing this. And for these first few genes, we are starting to see results and hopefully they will work. But I must also tell you that several of these trials have failed because it will be very difficult to show efficacy if there are so few patients who are involved with this, who are affected by the disease. Sorry, there's a cough coming. Excuse me. So being able to measure if a treatment is actually working in a relatively small patient group is a challenge in itself. And I'm going to discuss that a bit more later in the presentation. Now let's go to the next approach, which is really very hip and happening and the talk of the town. Many of you might have heard of it. It's called CRISPR-Cas. And CRISPR-Cas, that I would too easily try to explain it to you. It's very difficult, and the exact way it works is even very difficult for experts. But it's like a genetic scissors. It's not like the first treatment that I explained that you put in a new copy of the gene, but you clip out with some kind of molecular scissors. You clip out the defect, the problem in the specific gene, and then the own cell machinery repairs it in the good way, which is of course amazing. There are all kinds of pros and cons and downsides and advantages, but this is a revolutionary technique that will actually, I think, also be very important in the next few decades, I would say, for all kinds of diseases. And here on this photo, you see the two professors from Paris and the US who got the Nobel Prize for this invention, which is amazing. And then thirdly, I think I want to tell you a little bit more about that because there are new developments in this case too. There is RNA therapy. So I've told you about DNA. That's our genetic code. That shouldn't change, right? So that's the template of the genetic code. And then in the end, you want to have a protein. So our eyes, our whole body is made out of water, but also a lot of proteins. And the step in between RNA and between DNA and proteins is RNA. So you have DNA, RNA, and protein. And RNA is not stable, so it is an interim step, and it needs to be made time and time again by the cell. So, but this is also a way where you can also try to make treatments, and these are coming up too. Why is this important? Why don't we just do DNA gene therapy? Well, the reason for that is actually relatively simple. I talked to you about these virus envelopes, right? So these virus envelopes, just like a normal envelope, can only contain a certain size of gene. And many of the very common genes in retinitis pigmentosa, the most common is actually USH2A, which is not only the most common gene for Usher disease with retinitis pigmentosa and hearing loss, but is also the most common gene in retinitis pigmentosa. And this gene is far too huge to fit into this AAV. Now, the interesting thing is that quite a proportion of patients with abnormalities in this gene, they have a specific abnormality that you could actually target with RNA therapy. It's a very difficult story to explain how that especially works, but the gene is too big to use with the current methods with gene therapy, although there might be CRISPR-Cas in the future for this gene and other genes that are too big. But right now, we and others around the world are starting an RNA therapy trial where you get repeated injections every several months to half a year, repeated injections in the eye with a medication that actually corrects the disease at the cellular level of the RNA. And so this might be of interest to you. This will be the LUNA study, which is sponsored by Sepal Bio, which is a spin-off company of Thea Pharma. And this will be the first ever RNA therapy study for the most common gene in RP, which is USH2A. And we're actually going to start with the study in Amsterdam, for example, within a few months. And I know that around the world there are several centers who are going to take part. And I think this is going to be a very important trial. And likewise, we've also taken part in the trial for severe retinal dystrophy by the CEP290 gene, which I have also listed here, which was also an RNA therapy. But unfortunately, because of the small patient numbers and also maybe because of efficacy problems, these trials didn't make it to a clinical approval and the market, which is a problem for these rare diseases, of course. Other genetic diseases. So the previous genetic diseases that I've told you about are diseases where you want to correct the genetic defect. But there's another quite brilliant idea, although there are a lot of limitations, and that's optogenetics. So when these cells, these photoreceptor cells, are already dead, so the cones and the rods are not working anymore, what people have come up with is to express a new gene in the inner parts of the retina that are still working. So the outer cells are dead, but the inner cells are still there, in some patients at least. And what you do is you inject a specific gene that actually causes the expression of a new light sensitive protein. So the cells that are dead are not light sensitive anymore, but you make the inner cells light sensitive, which they weren't before, which sounds like a brilliant idea because you're bypassing the dead cells. So they have already tested this in humans with a certain degree of success, but this treatment will only be interesting and also, I would say, possible for people who already have advanced disease and who have a good inner retina. And still then it's not able to really bring back vision as it ever was, but it could improve vision to a certain degree. I do have to say that we really need more clinical data to really recommend this to more patients, because it is, first of all, a risky operation. Second of all, you also need a camera, as you can see on the right on the image if you're able to see it. So you need a specific camera. So more work needs to be done to see how useful it is. But this is another innovative approach, which can be actually categorized as gene therapy to bypass the cells that have died. Now, what if gene therapy is not possible anymore? And that brings me to the gene-independent treatments that I want to discuss. I cannot discuss them all. I mean, there's so much being tested, some promising, some less promising, and I want to discuss what I think are the more promising ones. So, gene-independent approaches. Well, for example, just to start, a lot of people are asking me when they come to my clinic, what about stem cell therapy? This has a magic sound. Stem cell therapy is when the cells are already dead, as in the previous example. The photoreceptor cells are dead, so the outer retinal cells are dead, but the inner cells are still good, which is not always the case, but let's say that's the case. For instance, it's quite commonly the case in retinitis pigmentosa or Stargardt disease, etc. Well, then, of course, it would be amazing if you can put in new cells under the retina. And that's what we and some others, there are only few centers in the world that are testing that, and we are one of them. We have a very good testing facility also for animals. We want to do animal testing as little as possible. But as you can imagine, of course, testing things in a petri dish is very different from in an animal. And before you want to take any risk in humans, you really want to first test it in other creatures. And unfortunately, we still need rats, and we're trying our very best, of course, to treat them as good as possible and to reduce the number of experiments, but this is still the reality. And what we found was that here you see below right, you see a picture of stem cells on a sheet. So a sheet with on top of it the cells were implanted under a rat retina, and this is done by Selina Costa and Peter Farnudian from our clinic. Well, this was done by Selina Costa, but we recently attracted a very good additional stem cell researcher from the US, which we are very happy with. But I want to emphasize that this is much further away before it can be implemented in the clinic than gene therapy, because as a surgeon, as an eye surgeon, I can tell you that it will be very difficult to put enough cells under the retina. You will need a bigger incision, and the cells have to be exactly at the right spot for them to work. So this is still quite a big leap, quite a moonshot to take. But we are trying to take the next steps to try to bring it to the clinic, and some other groups in the world are also working on that. And in my dreams, I would really hope that we could treat people with these hereditary retinal diseases, such as RP and Stargardt disease, with personalized gene therapy corrected own stem cells. But that still has quite some drawbacks because we need much more safety studies. And also, what's very important is that when your retina is degenerating, several layers of the retina are degenerating. So you don't need just one cell type. You probably need more cell types. These cells have to be exactly in the right spot to hopefully work. So we need more research to take this next step. This is also an interesting one. So this was stem cell therapy and gene therapy. Now we're talking about actually a relatively easy therapy that we are testing right now. I think this is one of the biggest studies that has been done to date and is being performed to date. This is the neck attack study. And neck attack is a study that tests just a pill. Oh, you have to take about 6 pills, I think, a day. This is a phase 3 trial, so it's not approved for the market yet, but we're testing if this is useful in people with retinitis pigmentosa. It is initiated by a university clinic in the US, Johns Hopkins University. And what they are testing, and what we are testing, because we're taking part in Amsterdam too, is N-acetylcysteine, which you can actually buy over the counter. I would not recommend it because it might also even be harmful. We think it might work, but we're not sure. So don't try it at home unless the results are there, I would say. Also because the purity of the N-acetylcysteine that you can buy online is often not very good. So what we are testing are these pills that might decrease the progression of the disease, or hopefully slow it down a lot at least. And this is, I think, the biggest study in RP ever done. And so we're in the middle of it. We just started with it like 1.5 years ago. I think there are almost 500 patients included from all over the world. We were very scared where Donald Trump got elected because he was downsizing the funding of the NIH, which is a sponsoring government institution from the US. But fortunately, we can still do the trial. But this was actually, everybody was really scared that they were also going to cancel this trial. But fortunately, we can proceed with that. And this also tells you that geopolitical developments can also influence science. So the neck attack study, and we expect to have results from this study, I think, in about 2 to 3 years. So that's going to be very exciting because here you don't need to have a specific gene to do the treatment. So if we cannot find a gene, or a gene is too rare to be eligible for gene therapy at the moment, if this would be shown to work, that would be amazing, of course. But let's see. Some of you might have also heard about these retinal chips, so chip implantations in the eye. So in patients who see very poorly, who have a very thin retina, which cannot be treated anymore, for instance, with gene therapy, but the optic nerve, the eye nerve, is still good. We and others have already tested the implantation of a computer chip in the eye. And this is, of course, also when many years ago, I was in Oxford for a year and I saw the first implantation of such a chip. And the patient who was not able to see anything, just a tiny bit of light, they switched on the chip and she could see shimmering and big forms, not more than that, unfortunately. But that was one of the most amazing things I've seen thus far. Although, big disclaimer, the company that actually brought such chip to the market has already stopped because the gain of vision is much too little. There's a high risk of the chip not working anymore, and the operation is very extensive. But still, I think it's hopeful that several of these approaches are tested, and hopefully in the future, we're still in the pioneering phase, might be implemented in the clinic. And of course, we all would like to halt the disease and treat the disease. But if this is not possible, I just do want to mention that digital support systems, if it's your mobile phone, but also small cameras on glasses, etc., such as this one, the OrCam, can also really help in at least supporting you in daily life, recognizing faces, reading print, reading barcodes in the supermarket. And this is still not working optimally, but hopefully, and I expect actually it to become much, much better because these digital developments in AI, etc., are going so fast and much faster, I must admit, than these stem cell treatments because you don't have to take a lot of risks. So hopefully, this will also improve daily life for many individuals. At least that's what I hope. So this brings me to the non-medical part of my talk, at least for a part. So the problem with these rare diseases such as RP and other hereditary retinal diseases is that they are not so common, right? So cancer and cardiovascular disease, they are very common. So pharmaceutical companies want to invest a lot of money in that because they can make a lot of money out of that. That's the real hard world, right? But it's not, big pharma is not good for rare diseases. So for rare diseases, we really need research institutions also outside pharma because pharma is not investing a lot of money in that because they cannot make a lot of money out of it. And that's why university clinics and research institutions all over the world, including our own in Amsterdam, are trying their best to really find treatments for these rare diseases, not only eye diseases, but also rare immunological diseases, rare cancers, etc., etc. So rare diseases need university research initiatives. And that's also because the funding for these rare diseases is much more limited because, of course, the more common diseases are more funded. And that's why I was very lucky, thinking about Frank and Dave. I'm going to show you a bit of more people who really took so much effort into philanthropic ways, so in funding, to be able to fund research for these rare diseases. For example, Dave and his wife Lineke, they started the Walk for Light, which was an initiative to organize walks, sponsor walks to get in money for this research. These are their beautiful daughters who are all three of them affected with retinitis pigmentosa, nevertheless doing wonderfully at school, at least the last time I heard Dave speak about them. And it's amazing to work with them on this fundraising. And here's another family, Rico family, who actually, with a village of 2,000 people, got 130,000 euros, more than that actually, together with all kinds of fundraising initiatives. Here's another family, the Petri family, who did an auction and raised a lot of money. Here's Rob Bruins, who was involved together with Dave and others in establishing our Eye Research Association in Amsterdam, which was officially signed here, to make sure that we can do our very best in developing these treatments. And we actually went to a musical together with many of us, celebrating our collaboration. And here's Roos Geltema, who also donated a lot of money to make sure that we can really take the next steps. And this is, I think, for the people involved, parents, patients, a good energy booster because you feel like you're really doing something about taking the disease further in terms of treatment. If it's not for you, it's for the next generation. And I must also admit that for me and for researchers who are involved in these fundraising programs and are supported by this, it is also a huge energy booster because you already have a very busy life, but this really keeps you sharp and motivated to bring it forward. because we are driven first and foremost by the people who we see in our clinic, and who we really cannot treat at the moment. And we really, for me personally, I really feel okay, I cannot sit still and see this disease degenerate. And also, I also want to be driven by my curiosity. So I really want to find out how the disease works and how we can really um, develop these treatments. And just shortly, how do we do this, right? So I'm talking about gene therapy, all these wonderful techniques, but how does that work in the clinic? How does it work in the lab? Because you need to do very extensive, time-consuming lab research done by wonderful, brilliant PhD students and postdocs. And how does that work? And this is what I call from bedside to bench and back research. So you start in the clinic with a patient who has a disease that you cannot treat and that you want to treat. And from that question, from trying to make a treatment for these people, you go to the lab and try to develop these new techniques such as CRISPR-Cas, etc., RNA therapy, and then try to bring it back from the lab to the clinic, which is the most difficult part. For example, here is a disease, X-linked retinoschisis, that I was intrigued about that we couldn't treat, and I fortunately got some funding to treat this. And what we on the upper part, there's a normal scan of the macula. Below, you can clearly see, you don't have to be an ophthalmologist. For those who can see it, there is an image where you can see that the layers are really disrupted, and we want to treat that. So what we can do, and I think this is really amazing, we can actually take we can make organoids, which are little organs in a dish, hearts, kidneys, but also eyes and retinas. And how that works is that we can actually now, which I think is a brilliant invention that started in Japan, you can take some blood or some skin cells, and with the right instructions, you can make those cells stem cells again. And a stem cell is able to go all the way to all kinds of different cells. It's a miracle cell. With the right stimulus, you can make a blood cell into a stem cell, and then the stem cell into a retinal cell or heart cell or kidney cell. So we, of course, do this for retinal cells, right? So we can actually take blood from a patient and uh, culture, make their own retinas in a dish, in a petri dish from that. And I think that's amazing because we can test all kinds of treatments in a petri dish in the retina that was made from the blood cells of the patient. So we try to do that as much as possible because we want to avoid animal studies. But still, as I also mentioned, or for instance, in the stem cell treatment, you really right now still need some animal models. And many people work with mice. We try to use rats because they have more complex visual behavior. And you can test these therapies also in rats. And we try to do that in parallel because we don't want to first test in the petri dish and then in animals, because then you will double the research time before you can get a treatment. So we want to do both at the same time. So a rat model of the disease and the petri dish model of the disease with the cells of the patient, him or herself. So we have a good animal facility where we can do that. And for this, of course, as you can imagine, you need a very good expertise infrastructure. You need PhD students in the lab. You need PhD students in the clinic who are going to show you, okay, these are the characteristics of these patients. And that is why these patients might be most eligible for the treatment. You need clinical people like me, but you also, of course, first and foremost, need to have brilliant people in the lab. And for decades, we are very happy to have an ophthalmogenetics lab that is, I think, really at the forefront in terms of developing gene therapy, stem cell therapy, which has been led for many years by my wonderful colleague Arthur Berge, who unfortunately died at the age of 62 very suddenly just 1 year ago, and we're missing him a lot. He was the head of our genetics lab, with whom I spoke every week, and where we developed new treatments. And we are missing him a lot because he was the captain on the ship when it came to development of these treatments. And we are still in the process of finding a successor to build on his legacy. So in these models, you want to find these new treatments, and then the big last step will be to bring those treatments to the clinic. And this is where I have to stress that this step right now will cost millions. So the lab research is already quite expensive. It costs tons of money. But then if you have a treatment and you want to translate it to the clinic, it costs millions because you have to show safety and make clinical grade treatment. So you really have to have very strong signals that it will work and have partners that are interested in investing into this last step. And below on the right, you see me doing the gene therapy surgery in one of the first patients who we treated with gene therapy in our clinic, which I think was one of the most amazing experiences in my life that we were finally able to do that. So this means blood, sweat, and tears, time, and money. So it is expensive, but it's also time consuming. So in the past, I've experienced that someone gave me some money to do research and said, okay, but I would like you to develop the gene therapy in 2 years' time. And then I said, well, you can give me 10 times the amount of the money, but we're not able to do that because we really have to go through the steps that are needed to show efficacy, and we have to use the right models. So it will cost time, but time is speeding up. So the Luxturna treatment that is approved for the market that I told you about, that took 20 years. Right now, it is possible to bring a treatment from the starting point where you do the first tests to the clinic between 5 and 10 years, which is still too long, I would say. Of course, you want to do it faster, but it's getting closer to 5-ish years before you can test the first people in trials. But then it still takes a few years to show in phase 2 and 3 trials that the treatment is actually working. Okay, so that's the reality. Uh, just to say that I don't want to paint castles in the air for you. I don't want to promise all kinds of things that we cannot deliver, right? So I think it's impossible. It's impossible to treat all of these genetic diseases, especially some of these genes that are extremely rare. But hopefully, in the future, in those cases, we can use other approaches, such as, for instance, N-acetylcysteine, if the NAC study will show that it is useful, right? So, what are the challenges? I think I'm taking a lot of time, but I think I'm still doing okay, I think, in terms of time. What are the challenges? Because I really want to be fair with you about the challenges that are still ahead, not only for us in Amsterdam, but all over the world. Because there are still some knowledge gaps that we have to bridge, and that needs more research and more funding. So I would like to talk about the challenges in terms of what, who, and how, right? So first of all, what? So we have to know for gene therapy, as I already told you, what the causal gene is. And I told you that from quite a lot of these forms of IRDs, we're not able to show the gene yet. So for instance, in recessive retinitis pigmentosa, we can find about 75% of the causal genes at the moment. So in these 25 remaining patients, they will not be eligible for gene therapy because they don't even know their gene. So in this case, we need to develop better techniques to find all the genes, and of course, other treatments if possible, right? So then another challenge, which is a challenge that has been overlooked and actually might be the reason why some of these gene therapy trials have failed, is who? Who should we treat? Who is eligible for treatment? Does the treatment actually work? Because several of these gene therapy trials have not been able to show efficacy. First of all, maybe because the treatment is not effective enough, but I wouldn't be surprised if it was because it was too difficult to show that it was working. And that's a waste of money because hundreds of millions can be spent for these trials. But if you're not able to measure well if the treatment is working, that's quite a waste of money. So first of all, it can be an ineffective drug. But there is also the question of, are we using the right tests to measure if the treatment is indeed effective, right? Sounds like a very easy question. Why didn't we think about it? Why didn't we do it before? Well, the reason is because these diseases are quite rare. So if you don't have the right measurements, you're not going to be able to show if the treatment is working. And one of the questions, silly enough, was how are we sure that the test is reliable? Because doing a test in healthy people, for instance, a visual field test, is one thing in healthy people. But in people who have an eye disease, such as retinitis pigmentosa, the treatment may be the measurement may be much less reliable, right? So, and is visual acuity or visual field testing or a quality of life questionnaire or a scan? What is the most useful way of testing if a treatment is working and safe? Well, that's why we went back to basics, and I think as one of the first, maybe the first group in the world, we did this research, which is maybe not as sexy as gene therapy, but right now we see it now that we have the results that we draw a lot of attention because people really realize, oh my God, we needed these data. So we wanted to find out who may be most eligible for gene therapy. That's why we did natural history studies looking at how fast does the disease progress and which measurements are most sensitive to show that the disease is getting worse. Because these measures might also be most useful to test if a treatment is working. So we want to know what are the characteristics of the disease and who are the patients who are most suitable for gene therapy and to show that it works, right? This is, of course, a logistical nightmare because you have to do all kinds of measurements in a specific patient and do them over and over again every year. And I think we are the first group ever to do a 7-year follow-up study for a specific form of retinitis pigmentosa caused by the CRB1 gene. And we are actually finalizing our 7-year follow-up, which is never done before. And that will be quite helpful, although it is a small group still. We need much bigger groups, but then we need money, of course, for worldwide studies to see which measurements are the most sensitive. And the second question I told you about, which is absolutely not trivial, is are these tests that we are doing in RP actually reliable? Because many of these gene therapy trials, again, spending hundreds of millions of dollars, didn't even know if what they were measuring was accurate and if it was reliable. So what we did, we looked at a group of 48 patients with retinitis pigmentosa with two main categories, poor seeing category and a better seeing category. These people did not receive any treatment, but we simply measured several things such as visual field and microperimetry. And then 2 weeks later, we did it again. Very simple, but nobody did it before to see what's the variability. And for instance, is the variability bigger in the poorer seeing patients than in the better seeing patients? It all seems logical, but we just didn't know. And interestingly, the FDA, so that's the Drug Approval Authority in the US, and in Europe we have the EMA. They actually had for instance, in retinitis pigmentosa and Stargardt disease, they said, well, for microperimetry, which is a very sensitive, specific visual field examination, you need at least 7 decibel change at 5 points to show that there's efficacy. Well, with our study that I just told you, we just now found out that it's completely unreliable. So this FDA measurement is completely unreliable. Whereas this is still now, by the way, in the rules. So I hope they will change that. Because what we found was that when you measure the volume sensitivity, this is quite technical. I will just go over it very quickly. But we actually measured that these 5 points didn't matter, was not reliable. But measuring the volume of the retina that can still see is much more reliable than looking at these points. So I hope and think also when others can also show that we were indeed seeing this correctly and finding this correctly, that this will actually change the way we want to test efficacy of disease. So we are refining the measures, but of course, a very important question I almost can hear you think, so what? Does it correlate with what really counts? And that's the experience of the patient, right? We can measure all kinds of fancy things with scans and visual fields, but does it matter to the experience of the patient? So nobody looked at that thus far. But we also started looking at that. And now the Foundation Fighting Blindness is also looking at that in specific cohorts. But the problem was thus far that there was not a good questionnaire for people with hereditary retinal diseases such as RP and Stargardt disease. The questionnaires that were out there that were validated were not useful for these diseases because they are very different from, for instance, cataract patients or age-related macular degeneration patients. But that changed a few years ago. The MRDQ questionnaire, the Michigan Retinal Degeneration Questionnaire, was developed especially for people with hereditary retinal diseases. And this questionnaire looked much more at functions in daily life that were affected in people with these IRDs. And they had several domains when it was looked at where you could score it. And then, of course, there's the question, does this questionnaire actually, and these domains, do they correlate with what we find in our clinical studies, in our measurements that we do in these RP patients? And fortunately, and this is going to be very important for future trials, I think, we found that the patient experience on these questionnaires correlated very strongly with a broad range of these visual function measurements and structure. So that's going to be quite pivotal when it comes to translating it to the clinical usefulness. Because, for instance, we can show, I would say, on a scan that gene therapy seems to be doing a good job. But the FDA and the EMA in Europe will only approve the treatment when it has functional benefit to the patient, and rightly so, because they will be expensive treatments, right? So this is really nice that we could, for the first time, show that there was a correlation. Of course, other groups also have to show it, but this is a good first step. And we're actually getting quite a lot of attention with these studies because they are so relevant for real life and for the clinic. So how do we do that in Amsterdam? And of course, it's not unique for Amsterdam in on other places too. I call it roof tile research, where the research of one student, one research question actually will expand and will enable you to go deeper and deeper with your knowledge. Because Mees Talib, on the left, there's a photo of Mees Talib, who was my first PhD student looking at this natural history study. And now I just told you we're at the 7-year follow-up. And Mees and Tan Nguyen and Leo Haan and Jessica Karuntu, who actually got a prize for research, Hind Amishata, Jonathan Hensman. We need really excellent PhD students, right? And these are all excellent PhD students. And Yasmin El Alali, they are really expanding this research and trying to deepen our knowledge to enable a better application of gene therapy. And actually, Yasmin El Alali on the right, she was actually hired with the fundraising by the Eye Research Association by OOG, with which we are organizing this webinar, and she's doing a great job. So to wrap up, is gene therapy going to bring us to infinity and beyond? Well, you probably already know that's not going to be the case. Some people will benefit from it. Some genes will benefit from it. Others not. Others might need different approaches, stem cell therapy, gene-independent approaches. So other challenges of gene therapy, because other challenges of gene therapy, which can make you ineligible, are that you have too advanced disease. You can put in a new gene in a retina that has no cells anymore. That it will not work because the cells need to be working to survive or to regain their function, right? So then, for instance, other treatments such as stem cell therapy in the future might be useful. Right now, we're only treating the center of the retina with an injection through the retina, so the macula. So we will not be able to bring back the peripheral retina with the visual field. And people will still have night blindness in many cases. But if we are able to save the center of the retina where the sharpness of vision is, there will be already a big gain. But I hope in the future that just a simple injection in as early as possible to save as many cells as possible with just an injection in the eyeball, so not an injection under the retina, will do the job. But that's not effective enough and has some downsides that we have to tackle. I think we need to do more because right now it will not work. The rare genes, I told you about that. If the genes are really, really rare, gene therapy will not be possible. And hopefully in the future, of course, when you have a super rare gene, if we have found out how we can easily adjust gene therapies to a specific gene, and we have already found that it is safe for another gene, we might hopefully in the future, and that's what I hope and what I dream of, be able to, even for a specific person with a very rare gene, try to develop gene therapy in a cost-effective way. But this will need time. Large genes are a problem, I told you. Some of them might be eligible for RNA therapy, as I told you with ASH2A and the LUNA study. But large genes may be eligible also for CRISPR-Cas treatment, as I told you, so the genetic scissors, because the viruses that we are using now, the virus envelopes, are too small. And for instance, in Amsterdam, we're also looking at non-viral ways of putting in the gene. So so you don't use a virus, but you use a kind of lipid particle to put the gene in, and that could also carry larger genes. But that again, of course, as always, has their ups and downsides. And finally, I would like to tell you something about the skeletons that are coming out of the closet. Of course, when you do research and when you treat people, sometimes you get side effects. And the talk of the town, which was Luxturna, so the first ever approved gene therapy, everybody was wildly enthusiastic about it. It was in some patients as if you put the light switch on again. But unfortunately, after a few years, it was found that actually in some patients, the disease was increasing in speed and that areas of thinning were occurring in the areas that were treated with gene therapy. And the big question is, of course, is this specific for this gene therapy for this gene? Or if this is also something we have to worry about for other gene therapies that we're trying with this virus and with this technique. So just to say that we're certainly not there yet, we have to be very vigilant, very alert to see if there are other side effects, and we have to change treatment approaches to make sure that we avoid this. What I can say is in the patients that I've treated with gene therapy in another trial, which the results haven't been published yet, thus far I haven't seen this side effect. So hopefully it's more specific to this specific gene, but we have to see. So there will be no miracles. We need hard labor to find new treatments and to be vigilant about potential side effects. But I do think that we are really headed towards personally tailored regenerative medicine. That's what it is, right? So the right gene therapy or stem cell therapy or other therapy for the specific disease phase and gene, because we want to make the right diagnosis in an expertise center to make sure that we give the right treatment to the right patient at the right time. That's our goal. And that's why we need several treatments to make sure that we have in the future treatment for everybody, which is now by far not the case, but that's what we're working on. So, all in all, we are in pioneering times still. They're exciting times, first times ever where we're trying these new therapies, such as gene therapy. And I hope that I have given you a glimpse into the new developments, some of the newest developments that are out there. I hope that I've also shown you that this is teamwork, requiring not only medics, but a very brilliant PhD students in the lab, in the clinic, postdocs, um, and also funding. And people are raising funds to make this amazing research possible. And it's an amazing time to work in. A question that I often get more and more actually, because we are doing quite some research and seeing a lot of patients, is from all over the world. People ask, "Well, can I visit your clinic because I've heard you're doing this and that?" I would recommend you, if you're not living in the Netherlands, to find an expertise center close to where you live, because all over the world, many, of course, uh, not in all countries, I must admit, but for instance, in Australia and India, uh, in America and Canada, and in Brazil, and all over Europe, there are very good expertise centers. So if you're on their radar, if you have had your genetic testing, if you have let them know that you are open to taking part in trials, you are on the radar. So you don't have to travel to the US or Australia or wherever, or to Amsterdam to be on the radar if there are new treatments. And in Europe, we are fortunate to have the European Reference Network for Rare Eye Diseases, in which I'm also active, where the European member states, many of them, almost all of them, have expertise centers for all kinds of rare eye diseases, including the rare retinal diseases, such as retinitis pigmentosa. So if you go to such expertise centers, you know you're on the right track. If you want to see, if you're from Europe, you're calling in from Europe, have a look at www.iranstripeeye.eu. You can see all the centers. There are also other webinars, etc., on there. And in other countries, you also have these kinds of networks. So rest assured, if you're at an expertise center, you will be on the radar for new developments. My philosophy when I talk to patients is be hopeful, but I want to underpromise and overdeliver. I don't want to promise all kinds of things that we cannot realize, but I hope that I've been able to show you that there are amazing things happening, that despite the fact that it's pioneering times, we are getting there, and we're bringing the first treatments to the market. And I hope this will also be hopeful for you. Just to end, so this webinar was offered by OOG, which means eye in Dutch, and which is an abbreviation of Oogonderzoekgenootschap, which is Eye Research Association, which is a fund within the Amsterdam University Medical Centers Foundation, Amsterdam UMC Foundation, which is really meant as a vehicle to really drive groundbreaking scientific research here in Amsterdam. And we, as OOG, as Eye Research Association, we have the goal of helping to finance this cutting edge research, which is so difficult to finance for these rare retinal diseases. It is run by volunteers, and all the donations go to scientific research with a specific focus on children with these diseases. We have a website. You probably all have all seen it because you registered for the webinar in this website. And if you would like to take part, contribute, or maybe think about fundraising or donate, I would like to refer you to our relationship manager, Mrs. Megan van Hegedus. Her email address is on the right. There's also a QR code here. It can also be found in our website. So please do not hesitate to have a look and have a think about if you might be able to contribute here or for an expertise center near to you. Everything is extremely welcome and helpful. So that was my presentation. And I know that there are quite some questions, and we will have some time now to answer those. And Frank and Dave will kindly coordinate that. And thank you very much for your kind attention. And let's go to the questions.
Thank you, Camiel, for your presentation. Actually, we received one question during your presentation. But to be honest, I think you answered a lot of things in that question. Do you also see that question as well?
Yes. Oh, let's
There's
see.
a question about are the promising technologies that can accelerate research in genetic therapy and be developing tech aids that augment visual data to the brain using data from sensory organs like touch.
So I must admit that this is not my field of expertise, but I think that new technologies such as AI, but also new network analyses that are being published will really help, for instance, to find the missing genes, but also the missing disease networks, I would say. And I hope that it will also accelerate our knowledge. We are also working on AI in really analyzing all these images that we are making and correlating with functional testing, because this hasn't also been done before. That could also help us again with refining the measurements and the sensitivity of measurements to be able to show with less patients, with these small patient groups, if the disease is progressing and if the treatment is working. So I think these techniques will be more and more important indeed.
Yeah, okay, thank you. Via the email, we also received a lot of questions, Camiel, and Dave and I had the commitment to select a few of those questions. So maybe I can give the word to Dave for the first question.
Yes, thank you, Frank. I hope you can hear me.
Yes, I can.
Yeah, okay. So we collected some questions that participants asked when registering for this webinar. The first question, how soon will these treatments will be available to the general RP population?
Yes, I think that's an important question. I hope that part of it has been answered by my presentation, because as you might have learned from my presentation, I don't think there's going to be a one-size-fits-all treatment. But I do think that more treatments in the toolbox will be available, and that it will depend on the individual whether or not you're eligible for the treatment. And some treatments will be available sooner and some later. For instance, some forms of gene therapy will be available sooner than RNA therapy, whereas, for instance, stem cell therapy is much farther away. When talking about the general RP population, one of those treatments that might be actually applicable to the general population, if shown that it's efficacious, is, for instance, NAC, so N-acetylcysteine. So that's the trial, the NAC attack trial that we're taking part in. So it would be amazing if we can treat all kinds of people with RP or other hereditary retinal diseases without having to target their gene. But for instance, just with a pill, for example. And that's what we're testing right now. So how soon it will be available depends on the treatment that is tested or the stage of the disease.
Okay. Thank you. I also see another question in the messenger from this presentation, and that's what if you are not accepted for a consult in the research institutes? And I think the background is, I'm guessing, for if you want to join a trial or something, if you are not accepted, what is your advice to do then?
Yeah, so well, for instance, in the Netherlands, the way it works is an ophthalmologist who is not in an expertise center will refer to the expertise center. And I can imagine that that differs between countries. So, and of course, when you live in Western Australia and want to go to Eastern Australia, it's very different than going from the south of the Netherlands to Amsterdam. But in the Netherlands, if you want to go to a specific expertise center, you can ask a referral by the ophthalmologist and say, okay, I want to go to that center. Can you refer me to that because I have this and this question?
Hmm.
So, for instance, we never refuse people for a consultation. What does happen is, for instance, that we see them once and then they go back to their local ophthalmologist because we cannot do all the checkups repeatedly at our clinic because we get so much referrals. But then you are on the radar, right? Um, in Europe, at least, I know that if you look at the ear and eye network, the website that I showed you, there are the expertise centers, and you can also show your ophthalmologist. You can show the ophthalmologist that you would like to go to that center if possible. I just see that someone was denied referral to Amsterdam UMC, so
Yeah.
um, let's keep in touch about that because that's not what should happen.
Make
But
yourself clear. Yeah, send an email.
yeah, you can send an
Yeah.
email to your ophthalmologist can always contact me because if you have an hereditary retinal disease, you can be seen.
Yeah,
Yes, you
okay.
can ask the ophthalmologist to email me. If you're your own ophthalmologist, you can ask to email me. But I want to stick to the general questions
Yeah.
because
Yeah, yeah.
this should be
Yes.
appealing to
Yes.
everybody.
Yeah.
There was a question via the mail. You see it on the screen. Which therapies are close to conclusion, Camille? What kind of vision can they sustain in RP patients?
Yeah, that's also a good one. So well, several of these gene therapy trials, for instance, the RPGR X-linked retinitis pigmentosa trial, um, that is in the middle of the trial. So several of them will be concluded within a few years. And what vision can they sustain in RP patients? Well, the hope is that, for instance, the gene therapy treatments can halt in the best circumstances they can halt the disease in the area that was treated, so which was the center of the retina. Because right now we inject under the macula, only in the center of the retina, which is so important for central vision. So that's where you hope that the disease will stop, or that the disease progression will slow down. But the truth is that we're actually not sure yet. We will not be able to improve vision in most patients. We hope to be able to save the cells that are still alive. And what I hope is that when we show that this is effective, we can treat much earlier, so we can save more cells.
Okay, thanks.
Okay, thank you, Camille. Is there a medicine against RP? What can I do? Take vitamins or other things?
Yeah, that's also a very interesting point. So I do know, that's good that this question is asked. I do know that some ophthalmologists prescribe vitamins, vitamin A or omega-3 or whatever. That is all unproven. And for instance, in the Netherlands, most of my colleagues, including myself, don't prescribe all kinds of vitamins, etc. The reason is that it's not proven, which sometimes it takes time to be proven, but the problem is that it might also be worsening the disease if you take too high concentrations of this and that, because the retina is already more vulnerable. So, and vitamin A has been taken in the past and prescribed for RP, but right now it has been shown, and there is quite some literature and also some discussion has been there, but the consensus is that vitamin A has not been proven effective for RP, and maybe for some forms may even be harmful. So the answer is there is no medicine at the moment for RP unless you are, for instance, eligible for a trial or for gene therapy. And hopefully, new treatments will come on the market when shown to be efficacious. But many of you might have already seen that if you Google, I would recommend you not to Google about treatments because you will find all kinds of charlatan treatments that have been advocated with electrostimulation, ozone therapy. Ozone is actually quite harmful potentially. Acupuncture, whatever, you name it. I mean, acupuncture is probably harmless, although expensive and not useful. But oh yeah, it was just a question about it. So I don't think acupuncture is useful. It's not harmless. If you want to spend your money on that, that's fine. But I don't think it will do anything. But yeah, so there are a lot of treatments, and sometimes I'm not sure if there are the right motives to offer these treatments to people who are desperate to do something about their disease. I wish there were effective treatments right now, but there aren't. So yeah, I wouldn't spend too much money on unproven things.
Thank you. Camille, I see also a question about cone-rod dystrophy. That's also with my son. It started with the cone and then after the rod. But cone-rod is the same as RP, isn't it? And
RP
and the
is rod
question,
cone dystrophy.
yeah,
So rod cone dystrophy means first the rods go down, then the cones, and cone
yeah,
rod means first the cones get bad and then the rods. So it's actually the other way around.
yeah,
And I saw the question popping up if NAC
yeah.
N-acetylcysteine might also be useful in those patients. Well, it's not being tested at the moment, but personally, I would think that if they show that it is useful in RP, the logical next step would be to test it in cone rod dystrophy, because it might also be useful there. But let's just wait until it also has been hopefully shown to be useful in RP. But that takes time, and as I've told you, it will be difficult to show
Okay,
efficacy in these rare groups. But fortunately, the neck attack study, there are almost 500 patients, which is really great.
okay, thanks. And can you tell me more about the retina transplantation? The way the new retina is grown from stem cells, the difference of the used membrane, and how does the transplantation going to be performed?
Yeah.
You
So
also see
retina
that question.
transplantation. So
Yeah,
people have probably heard about cornea transplantation. The cornea is the front of the eye. That's done a lot all over the world. But retina transplantation, meaning that you transplant a new retina in your eye, that's not possible. And the question if it's ever possible, probably not. So retinal transplantation is not possible at the moment and is still very, 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 very far away, if ever possible. But when it comes to growing retinas from stem cells, what I've shown you a bit from the organoids, so you culture the cells in a dish and put part of this retina of those cells back in the eye, that might be possible in the next, I'd say, decade or so. So yeah, so retinal transplantation is not possible, but mainly maybe in the future stem cells or organoid retina cells, hopefully.
Question number 5. Can you talk about the optogenetics and the different drugs under development?
Yeah, so I touched upon that in my presentation a little bit. So optogenetics is the expression of a new protein in the inner retinal cells when the outer retinal cells have already died. So you actually make the retina light sensitive again, right? So one main approach has been tried and published, um, but it's not on the market yet, and I think we need more proof to see if it works. I'm not aware how many drugs are currently under development in terms of optogenetics. We're not involved in that in those trials in Amsterdam, and there are not so many centers in the world working on that. But hopefully, it will be shown that it is worth the effort because again, it's difficult.
thank you. Gene therapy, Camille, how to alleviate the constant pain in the eyes.
Yeah, so I'm not sure what exactly is meant by this question. I assume that this is a
Yeah,
question about by someone who has painful eyes since gene therapy.
I think so. Yeah,
I think it would be very important to discuss that with your doctor, with your eye doctor, because I'm not sure if I'm your doctor or someone else with the gene therapy, but um, gene therapy in itself doesn't cause eye pain. So there might be There might be a problem with the eye surface, for example. So it's important to go to your ophthalmologist if you have pain.
Did AI make it more promising to find the cure sooner? And can AI speed up the process?
Yeah, so we already touched upon that in an earlier question from the chat, I think.
Yeah.
The question is undoubtedly yes. I am starting to become more enthusiastic about AI. My heart lies more into in some other fields, but I'm also involved now in AI initiatives, as I just told you, with regard to being able to more sensitively look at the disease, see the progression, etc. But more complex AI systems might even help in even better patient selection, but also in selecting um, better candidates in terms of gene therapy, not patient candidates, but also potential treatment candidates. So if you know more based on AI about how the disease works, what the complex networks are in which genes are working, this could help in finding new ways to test if the disease can be treated. So absolutely, undoubtedly, it will play an important role, I think. But it will not replace lab studies for sure.
No. Camiel, if you also see the questions that are asked at this moment, if you see if there is something interesting, please.
Let's see. I will have a look. Let's see.
Yeah. That's the last question of Imre.
Yeah, so N-acetylcysteine, but that's I would refrain from using that outside the trial.
Okay.
I would just wait for the results of the trial because these are high, these are dosages, and you always have a risk that it might not work or it might be harmful. So as frustrating as it might be, I would wait for the results of the trial because it's a very well performed trial.
Okay, thank you. What is the status of the clinical trials in genetic solutions, and how can we participate?
Yeah, so as I've told you, it depends on the gene. If there is a gene therapy trial, and for the genes where there is a trial, there are different phases of gene therapy or RNA therapy. So first and foremost, I would recommend you to make sure that you have had genetic testing in a good center. And if your gene is known, you could also look on clinicaltrials.gov. So clinicaltrials.gov, put in your gene and gene therapy. Then you can see which centers might be working on that gene therapy and with a contact person. So that could help. And apart from that, if you're lucky to live in a country where they have expertise centers, let's say in India, the LV Prasad Eye Institute, in Australia, you have Sydney, Melbourne, Perth, and in Europe, of course, you have the Ear and Eye Network where you can have a look. Sorry, been talking too much. You are close to the fire where things happen. And in the Netherlands, for instance, you have Amsterdam, you have Nijmegen, Rotterdam Eye Hospital, and there are two centers in the Netherlands where they do a lot of gene therapy research. We're in Amsterdam, we're the only one doing stem cell therapy research, but gene therapy research is done in Amsterdam and Nijmegen.
And in the US, what are good places in the US?
Yeah, so if you want to go to the US, I will not, it will be a big list. If you go to the Foundation Finding Blindness website, there you will see the centers that are involved actively in their network and their research. So Foundation Finding Blindness in the US will be a good website to take a look.
Thank you. What developments are there for RP2?
Yeah, so for RP2, which is a rarer cause of X-linked retinitis pigmentosa. So I told you about RPGR, which is the most common X-linked retinitis pigmentosa gene. We have gene therapy trials that have actually 3 gene therapy trials, including the one that we are involved in Amsterdam. They are being performed. Unfortunately, this is not the case for RP2. And you gave me the questions before, and I could have a look, and I saw that there is currently no gene therapy trial going on for RP2. So hopefully this will progress to a clinical stage trial. The problem here is that it is more rare. And that's why it's more difficult to start a trial and to find people and investors interested in doing that. So hopefully other approaches such as a NAC will also be tested in this gene. The thing is, fortunately, that if we show efficacy in one of the trials for RPGR, X-linked RP, this will definitely also trickle down for RP2 because this will be an important proof of concept and safety, I would say. I saw a question in the chat. What about Erasmus MC, which is another hospital in Rotterdam? Sorry for the non-Dutch people who are following. So they are also an expertise center for these diseases, but they don't have a trial center. But they are an expertise center with Dr. Tianens, and they are really good. But they don't have a clinical trial. They don't do clinical trials. So if there's a clinical trial in the Netherlands, we have a very strong network of collaborating centers. So people from Erasmus MC are then referred to one of the clinics where they do perform trials, such as Amsterdam, Nijmegen, and in some cases, or Rotterdam Eye Hospital.
And yes, thank you. When will gene therapy become available, and for whom? Will it also be available for people whose specific type of RP cannot be determined?
So there's a difficult one, right? I think I made it quite clear in my presentation that
Yeah,
that's a problem.
I think so. Yeah.
So if your gene is not known, then you cannot have gene-specific therapy. So that's a problem. That's where we have challenges left. So then we need gene-independent approaches.
Yeah.
Unless in the future we are going to find the gene with new techniques, and then hopefully that will be my dream. We can also do gene therapy for very rare diseases, and everything that is done for other genes that are being tested now in trials. It's not that it's not useful for you. So please be aware of the fact that it is still very, very useful for other genes that are currently not targeted because we are testing safety, we're testing efficacy, we're testing technical methods. So this all helps in bringing the field forward, despite the fact that it might not be your gene that is tested at the moment.
Thank you.
How are these gene independent therapies are really performing?
We don't know. The trial is still going. That's research. So the trial is going, and in a few years' time, we will know. That's the short answer. No disasters, though, thus far. That's for sure. I see those patients back. No, not a lot of strange side effects, etc. At least not in the neck attack study.
Okay, thanks. Yeah. What are the chances of an RP sufferer to receive personalized gene therapy for specific and rare, even unique mutations?
Yeah, so as already highlighted, that's right now very difficult, actually impossible. But hopefully, that's what I really hope. I mean, these rare diseases, in the end, the big pharmaceutical companies will not be very interested in bringing it to the market. So we will have to find a business model, I would say, where we can still bring this to the patients, right? So I hope that in the future, when we can show that certain techniques like CRISPR-Cas and gene augmentation therapy or RNA therapy is at least safe, and that we can bring it under the retina, that we might be able to also target these rare genes and will not have to go through all these new phases of testing again. I think that's what where we will be heading in the future. It has to be, otherwise many of these forms will not be treatable.
Thank you. One in Dutch. Hoe staat het? Let me translate it.
Want to translate it?
Yeah, that's also okay.
So how is the translation of how is it going with gene therapy for the CRB1 gene? Is there still financing needed? Well, the second question is superfluous because we always need financing. No, well, this is an important question you raised. So I told you in my presentation that we are actually, one of our focus genes is the CRB1 gene. This is actually the gene where we have the 7-year follow-up. The first time ever we have the 7-year follow-up, which we are going to publish. But here's an illustration of the problem. So we have this brilliant gene therapy scientist, Jan Wijnholz, who is actually now in Leiden, who has the treatment ready to test in humans. But before you can take the next step, you have to do purity testings and do some animal testing and some other steps to make sure that you can try it in the first humans. And that's a step that costs 7 million euros, which is extremely expensive. And that is a difficult next step right now. Hopefully in the future, the cost will go down, the requirements will go down because we can show that it's safe. And despite the fact that there are several relatively rich families affected by this gene, unfortunately, thus far it has been proven impossible to get these millions together to take the next step. And then you're in limbo because you have it almost on the shelf, right? The thing you want to test. You need these last steps. But before you can get the pharmaceutical industry interested to take it further forward, you need to take this first in human step. So that's the difficulty. And that has to change in the future.
Okay. I also saw a question about the NRL gene. That's quite specific, of course.
Several of the genes that are mentioned in the chat are rare to extremely rare. So it's difficult to go into them individually, but it's important that these people already know that gene. That's what I would like to say. And as long as you are known with an expertise center, keep an eye on clinicaltrials.gov.
Yeah.
Potentially other gene independent treatments. That's the most important thing you can do, and have a checkup every once in a while. Yeah.
Next question is about if your vision is so worse, you only see light and dark, and will gene therapy ever be a cure, or is it then too late? It's maybe also correlating if you are older or something. If you're an old patient, maybe you can tell us a little bit more about that.
Yeah, that's an important question. So the short answer is gene therapy will not be a cure in that case. And this not only it does not only hold true for RPGR, but also for other forms of RP. If it's so advanced that you only see dark and light, it might be useful for some people who have advanced disease to have this optogenetics therapy maybe in the future, but we need more proof about that. And apart from that, then you will enter a different field of approaches, which is also a bit further away. Stem cells, computer chip. Although we are still not advanced enough to really give that in a useful way, that treatment. So that's in short the answer.
Okay.
Okay, the extra question has already been answered. So I want to thank Camiel one more time. Thank you for your time and your very interesting presentation. I want to thank all the participants for your attention. And we will publish this video in a few days on our website.
And I want to thank you, Dave, for organizing this, and Frank. You were amazing, and it's such a pleasure to work with you on this.
Yes, it's for the good goal, of course, and you're helping us also with your information. And we are very lucky we have you, Camiel, as our doctor, so we get a lot of information for you. But for us, it's very important that we can also share knowledge to the other people around the world who don't have that kind of entrance we have. So thank you for your time and that you're helping all of us with RP. Thank you very much.
You're welcome. All the best.
Okay. Bye-bye.
Bye-bye.
Bye-bye.